function of a synapse depends on neurotransmitters and receptors. Synapses come in two types. One is an electrical synapse. In an electrical synapse, gap junctions conduct a signal directly between the cytoplasm of two cells. We find electrical synapses in cardiac muscle, smooth muscle, and some neurons. The advantages of an electrical synapse are faster communication and synchronization. Chemical synapses are more common in the nervous system, and these are what we will be focusing on. The first neuron, also called the presynaptic neuron, releases a neurotransmitter onto the second neuron, also called the postsynaptic neuron, that responds to it. The number of synapses on a postsynaptic cell is highly variable. We may have 8,000 synapses on a spinal motor neuron, whereas we may have 100,000 synapses on a neuron in the cerebellum. The cerebellum is responsible for comparing our intended motion to our actual motion and for keeping our balance and posture. So the cerebellum gets a lot of input it has to process. Synapses can be axon to dendrite, axon to soma of the postsynaptic cell, or axo-axonic. Let's take a look at these. Axodendritic synapses are the ones we commonly think of, where the axon of one neuron synapses with the dendrite or receiving end of another neuron. An axosomatic synapse is when the axon synapses with the soma or cell body of the postsynaptic neuron. And finally, axo-axonal synapses are when the presynaptic axon synapses right along the postsynaptic axon. And we can have this either near the axon hillock, or we can see the axo-axonal synapse where the axon of the presynaptic neuron synapses down near the axon terminal of the postsynaptic axon. Neurotransmitters were discovered by Otto Loewi, and he demonstrated their function with an experiment in frogs. Previously, histological observations had revealed a gap between neurons. We call this gap the synaptic cleft. However, somehow these two neurons are still able to communicate with each other. So Otto flooded the exposed hearts of two frogs with saline and then stimulated the vagus nerve. The vagus nerve is the 10th cranial nerve, and it supplies most of the viscera. When it is stimulated, it is a parasympathetic input, and it will slow down the heart. So the first frog's heart slowed down. He then sucked the saline off of that frog's heart and found that it slowed down the heart of the second frog. So there's some chemical that was released into that saline that would then slow down that second frog's heart. He named this vagus substance, which was later renamed acetylcholine. We've examined synapses previously when we discussed muscle. The chemical synapse structure is the same as the neuromuscular junction, except instead of having a muscle on the other side of the synapse, we have a postsynaptic neuron. So our presynaptic neurons have synaptic vesicles with a neurotransmitter. It may be acetylcholine or it may be a different neurotransmitter. And our postsynaptic cells must have receptors. Here's a micrograph of multiple synaptic knobs, all synapsing on the soma of a postsynaptic neuron. This demonstrates how many synaptic knobs actually synapse on that postsynaptic cell. The effect of a neurotransmitter at a synapse is highly dependent on the receptor type. and we have different types of receptors. At the neuromuscular junction, we saw ionotropic or channel-linked receptors. These are ligand or chemically gated ion channels that open an ion channel when the neurotransmitter binds to them. At the neuromuscular junction, we saw excitatory ionotropic ligand-gated ion channels. So we also can have inhibitory ionotropic receptors that will let chloride or potassium through that channel, and that will cause hyperpolarization of the postsynaptic cell. The other type of receptor we have is metabotropic. These metabotropic receptors are linked to G proteins. This means there is no channel in the receptor, but the G protein or a second messenger may open a channel. These metabotropic receptors are generally linked to potassium channels and are therefore inhibitory. So here's a diagram of ionotropic receptors, and you see that normally the channel is closed. When the ligand or chemical neurotransmitter binds, the channel opens up and ions can flow in or out depending on their concentration gradient. When acetylcholine binds to an ionotropic receptor, it often opens a cation channel, which means that cations like sodium, potassium, and calcium can move through that channel depending on their concentration gradient. This generally creates an excitatory postsynaptic potential, an EPSP, because sodium moves into the cell. If instead we use the neurotransmitter GABA and it binds to an ionotropic receptor, it opens a chloride channel. So chloride is able to flow into the cell, and because chloride is negatively charged, it will hyperpolarize the cell and cause an inhibitory postsynaptic potential, an IPSP. Metabotropic receptors are linked to a G protein. The G protein is our little second guy in the little relay race where we're passing off the baton. And that G protein can either open a channel directly, so this G protein right here at the bottom can move over and stimulate this channel to open. In this case, it's a potassium channel, and potassium will flow out of the cell, creating an inhibitory postsynaptic potential. We can also use that G protein, as you see in the middle figure, to activate an enzyme, adenylate cyclase, and create our second messenger, CAMP. CAMP can then go to a ion channel and open up that ion channel so ions can move through. Depending on the cell, we can open ion channels, close ion channels, and therefore may create an excitatory or an inhibitory postsynaptic potential. Some of the neurotransmitters that we're going to use to stimulate these ionotropic and metabotropic receptors are going to be nitric oxide. Nitric oxide has an indirect action. That means it doesn't directly open an ion channel, but it goes through a metabotropic receptor. So indirect action is metabotropic receptors. It's an excitatory gas which diffuses in the brain, and it acts as a vasodilator in blood vessels. Acetylcholine is a neurotransmitter we've seen before, and it's formed from acetic acid and choline. Many of our peripheral nervous system neurons release acetylcholine, and some central nervous system neurons release acetylcholine. Depending on the receptor on the postsynaptic cell, it may have a direct or indirect action. At ionotropic receptors, that we also call nicotinic receptors, acetylcholine is excitatory and opens cation channels, allowing sodium to move through. This is the type of receptor and neurotransmitter that we saw at the neuromuscular junction. Acetylcholine is usually inhibitory at metabotropic receptors because it will open potassium channels. Acetylcholine is inactivated by degradation by that acetylcholinesterase enzyme. We also have amino acid neurotransmitters. These are amino acids which are released and bind to receptors in order to cause a excitatory or inhibitory postsynaptic potential. Glutamate is one of our excitatory amino acid neurotransmitters, and it's found in the brain and central nervous system. It triggers an ionotropic receptor for sodium and therefore excites the cell. It is inactivated by reuptake into the synaptic knob by transporters. So we have special transporters in the membrane of the synaptic knob that can basically pick up that glutamate and bring it back into the axon terminal. We also have two inhibitory amino acid neurotransmitters. They are GABA and glycine. These both bind to ionotropic receptors that allow chloride to pass through the ion channel. These are only found in the central nervous system. We also have biogenic amines, which are synthesized by replacing the carboxy group in amino acids with a different functional group. We have our catecholamines, which include epinephrine, norepinephrine, and dopamine. And in the brain, when these are released, they deal with emotion, mood, and pleasure. These all function through an indirect action, which means they use metabotropic receptors. And because we have different receptor types, these neurotransmitters may be inhibitory or excitatory. These neurotransmitters are inactivated by either reuptake into the axon terminal or degradation by an enzyme called monoamine oxidase, MAO. Serotonin is our other type of biogenic amine, and we find this in the brain and central nervous system. It has an indirect action through a metabotropic receptor, and it generally is inhibitory. It's important in sensory perception, mood, and sleep. Neuropeptides can be excitatory or inhibitory, and they work at metabotropic receptors through an indirect action. These neuropeptides act at lower concentrations than most neurotransmitters and have longer lasting effects. These are generally chains of 3 to 40 amino acids, so they're larger than our other neurotransmitters, and they're stored in the axon terminal as larger secretory granules. Some of these are also released from non-neural tissue, like the gut. So these gut-brain peptides are one of the reasons for food cravings. Some of these neuropeptides also function as hormones. They're transported through the bloodstream and modify actions of other neurotransmitters. Next, we'll examine the relationship between the neurotransmitters and receptors and how that makes a functional synapse.